So our final talk is from Dr. Lea Starita. So Dr. Starita is a research assistant professor in the Department of Genome Sciences at the University of Washington and the co-director of the Brotman-Beatty Advanced Technology Lab. Her lab has applied multiplex analysis of variant effect to genes important for understanding cancer risks, such as BRCA1, and genes important for understanding drug metabolism, such as TPMT. Her current goal is to develop the next generation of multiplex analysis for variant effect with richer, more informative phenotypes and the ability to assess complex genotypes. So welcome, Dr. Starita, and please take it away.
Thank you. I'm just looking for here it is, I think. Yes, okay. So thank you for inviting myself to talk, I guess, today. Okay, so I'm going to talk to you about the promise and progress of MAVE for clinical variant interpretations. This is going to be a tiny bit of history and plus where we're trying to go in using this data in clinical variant interpretation. So one of the promises of the Human Genome Project and now all of these precision medicine initiatives is that we're going to be able to use the personal genome sequence of individuals to understand their disease risk and maybe response to treatment and develop individual responses or individual treatments for people. However, we are way better at sequencing or reading the human genomes than we are at understanding what we find there. And this donut plot shows the contents of the clinical genetics database ClinVar currently. And this is just for single nucleotide variants that cause missense, single nucleotide variants that cause missense or amino acid changes. And you can see that the overwhelming majority of these variants are variants of uncertain significance. And this is the 1% of the genome that we know the most about, right? This has nothing to, non-coding genome is, way worse. And unlike the more definitive categories of benign or pathogenic, VUS can't be used for clinical decision making. And they just can't do anything with these results. And you can imagine that VUS are a very frustrating result for both the clinicians and the individual tested for disease. It had been conventional wisdom in the field, I think, that as more sequencing was done for these big genomes, genome studies or diagnostics, that this problem would go away, right? With more sequence data, we'd be able to infer more connections between the variant and disease risk. However, this is really not the case, right? So the VUS are actually increasing exponentially without really much concatenant change in the more definitive categories, right? There is this little bump in benign, and I have a feeling, I think this is probably from NOMAD and the big studies, NOMAD and Bravo, which show that some variants exist in the population at a higher allele frequency than could possibly be causative, but otherwise, these other categories really aren't moving at all. And this, this has real world consequences, right? So a lot of the genes that are in ClinVar are what we call clinically actionable, which means that a VUS, or meaning that if a variant is known to be pathogenic, we can the disease risk for the pathogenic variant can be ameliorated by medical management, right? And actionable is actually a very specific term that is used by the clinical genetics field. BRCA1 is a prime example. Women with pathogenic variants have a 50 to 70% lifetime risk of breast cancer, but risk reducing surgery can greatly, can take that risk back down to the population level. And you can see that in this Kaplan-Meier curve of women with BRCA variants that either had risk reducing surgery or did not. And obviously that makes BRCA1 and BRCA2 very well studied genes. And you can see that even in this extraordinarily well studied gene, most of the variants are variants of uncertain significance. So how are we going to solve the variants of uncertain significance problem? Well, you all are at the mutational scanning symposium. So, I'm going to suggest that is going to be the answer. So I'm going to give kind of a generic introduction to some of the talks we've seen here and a lot of the talks that we see in from our own graduate students in genome sciences. And that is that, oh man, disease X is caused by gene Y. And we keep sequencing this gene and we keep finding these variants of uncertain significance, but I can't tell if these variants are going to be pathogenic or benign, or what this variant is going to do to the structure of the protein. Is this variant going to allow this protein to fold nicely, or is it going to be unfolded and cellular equivalent of garbage? And so we can actually measure the effect of each of these variants on the biochemical or molecular function of the protein, and we can try to figure out what that does to the cell. But doing this one by one takes forever. So obviously, if we link this up to the powerful next generation sequencing, we can multiplex these assays and we can make a lot of mutations in all of these genes, right? And so what we have to do is now scale this up and figure out how to do this for more genes and more phenotypes. And so multiplexed assays have come a long way. So the first multiplex assays were really done in 2009 and 10. This is actually a figure from Doug's paper of 25 amino acid WW domain. But there's been a lot of tech development to actually getting to where this data can be used in the clinic. So this is actually Doug's first paper from 2010 in Nature Methods. And we could see that from, we could get really fine grained information of what, every single variant in this gene. And we figured, if we could scale this up and we could apply it to clinically actionable genes like the ACMG59 or pharmacogenes or cancer drug targets, we might be able to build an atlas of variant effects, right? And we could develop more, more accurate assay systems and access more diverse phenotypes. So the first proof of principle we published was this scan of the BRCA1 ring domain. So we developed two multiplexable assays here. First, BRCA1's N-terminus is a E3 ligase, E3 ubiquitin ligase ring domain. And so we had a phage assay to determine for that. And then it has to bind this other protein called BARD1 or it doesn't fold. And we had a yeast-2 hybrid assay. And combining the results from both of these deep mutational scans, we were able to actually fairly well separate known benign and known pathogenic variants that overlap this data set. Again, this is a cDNA assay, so we couldn't do anything about splice variants. This variant just wouldn't behave. I still know what it is. It was L22S. But for the most part, we could see that some variants were not going to have a great effect on the protein, and some caused, were damaging to the protein. But even though this assay wasn't, perfect or clinic ready, what we could determine was that these scores from multiplex functional assays were better at predicting the functional effects of the full-length protein than commonly used variant effect predictors. So we thought, okay, well, this is really worth following up. And in the last couple of years, many more mutational scans on human genes. We're using more and more advanced systems. And this is not all of them, but there's a lot of data here. We've been able to do tricks to barcode genes and be able to look at longer sequences. We've been editing to install the genes or install the mutations right into the genome to look at all sorts of different phenotypes, whether it's drug sensitivity or survival. And Jochen Wheel and Fritz Roth at the University of Toronto had counted up that about 250,000 variants have been made and measured by about 2 years ago, and I'm sure that that number has almost doubled by today. But one of the things we really haven't answered with this data is how useful is it for clinical variant interpretation, right? We keep saying that we want to do this, and that I keep telling the NIH that this data is useful and we should be producing more of it, but we really haven't quantified just how impactful it could be. So we set out to answer this question using multiplex functional data for 3 cancer susceptibility genes, and each presented different challenges. First, for BRCA1, this is actually a fairly simple story. This is the saturation genome editing data measuring the depletion of functionally abnormal BRCA1 variants in a haploid cell line. And this data is from this paper at Finley from 2018. For P53, this is a more complicated story, right? So P53 can be loss of function. It can also be dominant negative. And so all of this data came, from the Hahn and Ebert labs at Harvard and Dana-Farber. This is about 8,200 variants. And then for P10, we use the 2 data sets, the functionally abnormal, or the variant abundance data set and phosphatase activity data set. This one is from the University of Washington, and this one from the O'Rourke lab at OHSU. And what you can see about this one that's going to be weird, right, is how few known benign variants are in this data set. And this is actually going to cause us problems later on that I'll explain in more detail. Okay. And so the work I'm going to tell you about was done by Sean Fayer. So Sean Fayer is a graduate, is an actual genetic counselor who was so frustrated with the VUS results that he came across in the clinic that he decided to do something about it. And he is now a genome sciences PhD student that is co-advised by myself and Doug Fowler. So this work is led by Sean, and who actually you saw this morning. Okay. So the first thing Sean did was to curate the data from these publications to assess the quality of the data sets and whether or not they could be used for this exercise, right? And he did this to guidelines that we wrote to assess multiplexed functional data sets last year. He assessed things like dynamic range, the error associated with each variant. And I'm going to use this opportunity while I have you all here to ask you people to use HGVS nomenclature for your variants so that your data sets can easily be compared to genetic data. And please maybe read this paper so that we start getting data sets published in a more uniform way. So once he had gathered and assessed all of the data, he could move on to assessing the strength of evidence that could be applied to variant interpretation. The strength of evidence that can be applied will determine how much weight the multiplex functional data has when it's combined with other evidence for or against pathogenicity, right? And so strong, moderate, and supporting aren't terms that Sean made up. These are actual real terms from the the ClinGen genome resource. And the way we determine the strength of evidence is by calculating the odds of pathogenicity, or odds path, as recommended by the ClinGen sequence variant working group. Okay, and in this paper, the ClinGen sequence variant working group really set out to nail down how to use functional data in variant interpretation. And so we followed the guidelines in this paper. And so the odds path is a Bayesian framework that quantifies how well an assay is able to discriminate known benigns and known pathogenics. And the more an assay gets right, or in each of those classes, the stronger the evidence can be, right? So this BRCA1 data set has very high sensitivity and specificity, such that we can use strong evidence for pathogenicity and strong evidence for benign. And you'll also notice that there are a lot of known pathogenic and known benign variants for BRCA1, and this is gonna be not the case in another gene at the end here. So the third step then is to take that evidence and apply it using, we're going to again use a Bayesian interpretation framework here, but you have to add that to other evidence for or against pathogenicity, right? Functional data alone is not enough to reinterpret a variant. So where are we going to get this other existing evidence, right? Unfortunately, publicly available databases are incomplete and not useful for this kind of exercise. So we collaborated with Ambry Genetics and their amazing variant interpretation team, Tina Pesaran, Carrie Horton, Holly LaDuca, and Rashid Karam. And they very generously gave us all of the information that they had for all of the variants that overlapped the BRCA1, P53, and P10 data sets. And you'll notice that for each of these data sets, there are a lot of variants of uncertain significance in each of them. So first, so we're going to look at BRCA1 first, and Ambry had not taken a full advantage of their own data, like family history especially, or the updated rules for variant interpretation. So Sean did that first, and without using any functional information. And he showed that by just doing that alone, he could reinterpret 24% of the variants of uncertain significance, which left 76. Adding strong evidence both for and against pathogenicity using the Bayesian variant interpretation framework, he showed that he could reduce the number of VUS to 14, which is actually an 82% reduction in VUS, which I thought was actually pretty amazing. And again, the cool thing about these data sets is that they are future proof, right? So the next time Ambry finds another variant in this region, we can go back and kind of do this exercise again. Okay, so next we wanted to assess the impact of the TP53 data from the Hahn and Ebert groups. And since the P53 data is more complex, right, you can't draw a line on any one of these data sets and really separate the pathogenic and benign variants. And you want to consider the loss of function and the dominant negative effects. What Sean did was he built a naive Bayes classifier to predict the functional effect. And we built it predicts whether or not the P53 is going to be functionally normal or functionally abnormal. And again, these terms that I'm using, functionally normal and functionally abnormal, are actually developed as an ontology by ClinGen. So I'm not just saying these, they sound a little funny, but it's actually an ontology developed by ClinGen. And so the classifier performs fairly well at very well at identifying functionally abnormal variants, and he can use the functionally abnormal variants as strong evidence for pathogenicity. But it misclassifies a few pathogenic variants as benign. So this data is now capped at the moderate evidence for benignity, which I'm going to call that a word. Okay. So how did this work? So how well did this work? So again, there were 166 VUS in Ambry's data set. There was using strong evidence for pathogenicity, moderate evidence for benign, and then the expert panel interpretation framework, plus with the exception that we're going to use why does this keep doing this? With the exception that we're going to use all the multiplex functional data instead of the limited set that the expert panel allows. We showed, actually the expert panel wrote the guidelines before some of this data was published. So we show that we're actually able to reduce the number of VUS from 166 to 49, which is a 70% reduction in variants of uncertain significance. Okay. So that was, I think that pretty strongly shows that this data can be very useful. But what happens for P10, right? So this is this last data set. And we've got a problem, right? There's not a lot of benign variation in P10. I guess you really don't want to change this gene. And so therefore, we really can't assess the assay specificity. And so therefore, we're limited to what we can do here. So the expert panel for P10 says that you can use this data as supporting evidence if it's found to be functionally abnormal in both of these assays. And so Sean was able to use that information to reclassify about 15% of the variants of uncertain significance. And now, in this story, 2 out of 3 of the genes had enough known variants of known effect. But it turns out that a lot of genes, especially even ones with their, there's really well-known effects of these genes, and they're in the ACMG 59. Only 15 of them could you get to have enough gold standard variance for benign and pathogenic that you could actually get to strong evidence even if you had a perfect MAVE. So if you had even if you had a perfect MAVE for most of these genes, you would be capped at supporting or even no evidence, not being able to use this evidence at all for 18 of them, which is not good. So in order to maximize, so one of the challenges, I think this is one of the challenges that the clinical variant interpretation workstream of the Atlas of Variant Effects will take on. And I think we really want to see the multiplex functional data used to its maximal effect. Okay, so we think this data is still worth creating, even if sometimes it's going to be hard to validate. And so we are scaling up at the Brotman Beatty Institute Advanced Technology Lab. So we're building a centralized production facility for these types of assays. And we're actually hiring, I'm going to shamelessly plug here, a director of mutational scanning. So search Brotman Beatty at UW Hires for that position if you're interested. Another thing, I'm part of a group called the CMAP, so the Center for Multiplex Assessment of Phenotype, with Doug and Fritz and Judith Vienne, Christine Quaich, Stan Fields, and Jay Shanduri, and Cole Trapnell, where we're trying to unlock new phenotypes, unlock access to new phenotypes using new assays. And we're actually hiring postdocs here. And again, the Atlas of Variant Effects, we're trying to propel more people to produce more of this data and to really get together and figure out how to use it to make it maximally impactful. And there were 130 members when I made this slide. And just a few minutes ago, Lara Muffley, who is our fearless project manager, said we have onboarded 163 new members during this meeting. So that's really exciting. So thank you very much, and I'd love to take questions.
Great. Thank you, Dr. Starita, for the excellent talk. I'm sure it's very valuable to many people here. We can go to questions now. So first question comes from Claire Turnbull. What did you do about the IARC Cato transactivation assay? The ClinGen panel put more weight on the Cato than the Giacomelli. I'm hoping I pronounced that right.
Giacomelli, yes.
Giacomelli.
We actually found, so obviously there's a lot more data in the Giacomelli data set, and that second set, the second set actually, I think it's the same library from Botcher et al. And so we actually showed that the multiplex functional data sets, so these newer data sets, have better AUCs, so better ability to predict pathogenicity or better ability to predict functionally abnormal and normal variants than the Cato data set. So we actually ignored it, which I know we're not supposed to do. But in this data set, we were specifically asking about the power of the multiplex functional data sets. So we put the Cato data set aside.
Great, thank you. So next question is from Amy. They ask, is there a funding mechanism or a large-scale initiative to support interaction between technologists and clinical geneticists to do variant reclassification following MAVE studies?
There is an RFA. Fritz Roth showed it to me the other day. It does look like there is one, actually. Maybe if Fritz is listening, he could post it in the chat. I did see one. But I'm hopeful that IGVF, so that the new NHGRI impact of genetic variation on function, I'm hopeful that there will be a little bit of that funded there, or at least after some of the data is produced.
Awesome. So next question is from Fritz. He says, is it common in judging computational predictors, for example, by precision versus recall or RC analysis, by aggregating info across many genes? Are there any ClinGen activities thinking about aggregating like genes and MAVEs to allow for the calibration of evidence?
Not that I know of. You know, somebody could set me straight, but I don't think so yet. I mean, that could be something that we do for with the clinical variant interpretation working group through AVE, though, and in combination with Debbie's group. That would be really neat. The VTMST. Sorry, I'm not even going to go there. Go ahead.
And yeah, so the final question for you is from Aisha. So she asks, given the limit in population plus segregation data for rare variants, what do you think is needed apart from really good MAVEs, like the one you showed for BRCA1, to reclassify the 14 VUSs that weren't reclassified using Bayesian classification from Findlay et al.?
That is an excellent question. I really haven't dug into the data, so I don't know what is underlying that. It could be that certain other pieces of evidence maybe don't agree with each other and are canceling each other out, or that the data doesn't exist. I actually don't know the answer to that. So maybe if Sean Fayer is listening, he might be able to answer that question in the chat.
Great. So thank you very much, Dr. Starita. I'm sure you can continue answering questions in the chat.